Hello, I am Evan Shadrick. For my final presentation, I chose Dr. Bruno's Seafood Forensics Lab. Dr. Bruno is a marine ecologist and works with a group of students every year to quantify mislabeling rates of various types of seafood. I chose this lab because I'm particularly interested in the research being conducted and its potential impact. For instance, I believe that the natural environment should be maintained. Seafood mislabeling puts this at risk because it sends an inaccurate signal to consumers that a specific type of fish may be more abundant than it actually is. This has the potential to lead to overfishing and exploitation of the resource. Additionally, the inaccurate labeling of seafood puts consumers at risk of things like an unintended allergic reaction or even mercury poisoning in extreme cases. The scale of mislabeling and lack of regulation also surprises me. The final reason I chose this lab was the experience it allowed me to gather in things like DNA sequencing and field sampling. I was one of Dr. Bruno's students last year. The aspect of the lab that most interested me was the purpose of the research. The research worked to bring awareness to the prominence of seafood mislabeling. I intend to highlight that aspect with one of the lab's research publications. The article is called "A High Proportion of Red Snapper Sold in North Carolina is Mislabeled." In this article, 43 samples were collected from grocery stores, restaurants, and seafood markets. The DNA sequence of the red snapper samples was then determined using Sanger sequencing, then BLAST. I will discuss these in more detail later in the presentation. Ultimately, it was found that 90.7% of the 43 samples were inaccurately labeled. On the bottom left is an example of what a red snapper looks like. The researchers hypothesized that a significant number of the samples from each of the three vendors would be mislabeled. This initial thinking stemmed from the high price of the fish and the unusually short harvesting season caused by overfishing. This worked to reduce the red snapper population. This is the methods and experimental design. These two things are the same for all of Dr. Bruno's research related to determining the mislabeling rates of seafood. The first step of his laboratory techniques is field sampling. In the context of this research paper, one sample was collected from each of the vendors. And the vendors include 14 grocery stores, 12 fish markets, and 17 restaurants. The second step is, of course, the DNA sequence of all these samples. This was done using PCR, which stands for polymerase chain reaction. PCR works to make millions of exact copies of specific DNA sequences predetermined by the researchers. Gel electrophoresis was then done to determine if the DNA amplification caused by PCR worked properly. This newly created strand of DNA is then hit with a laser, which makes one of the nucleotides glow a certain color depending on whether it is A, T, C, or G. A light sensor then records that glow and transmits it to a computer, which orders the base pairs into a chromatogram. The third step is the use of BLAST. BLAST is essentially a massive database with the genetic sequence of thousands of organisms reported. When you blast a sequence, you compare it with all of those organisms in the database. If the nucleotide sequence matches, then the organism is identified. The fourth step is statistical interpretation and determination of significance. In Dr. Bruno's lab, this is typically done using a chi-square test. Then this is just a visual representation of what is occurring in different ways to describe Sanger sequencing. The three main steps are: number one, PCR with fluorescent chain terminating ddNTPs. Number two, size separation by capillary gel electrophoresis. And number three, laser excitation and detection by sequencing machine. The results will be used to bring awareness to countless people. In the red snapper paper, 90.7% of the samples were mislabeled. 23% were within the same genes, 58% within the same family. My personal take on this paper is that it is safer to buy from grocery stores, as only 71% of the grocery store samples were mislabeled, while all of the samples from the sushi restaurants and the fish markets were not red snapper. Some future advancements that may result from the research in this lab include the potential to create a database allowing consumers to make informed dietary decisions. This database could include things like the mislabeling rates of restaurants versus grocery stores in various cities across the United States. This is just a reference slide. Thank you for listening to my presentation.